Welcome, everyone. This presentation walks through the science of hormones, the chemical messengers that coordinate virtually every function in the human body. We'll cover three interconnected topics: the chemical structures of key hormones, how they travel through the bloodstream to reach their destinations, and what happens when they arrive at their target organs. By the end, you'll have a clear picture of why a tiny molecule secreted in the adrenal gland can make your heart race within seconds, while a steroid hormone produced in the gonads can remodel your bones over years. Let's start with the definition. A hormone is a chemical messenger secreted by an endocrine gland directly into the bloodstream, where it travels to target cells elsewhere in the body and triggers a specific response. What makes the endocrine system different from the nervous system is distance. Nerves send signals along dedicated wires. Hormones broadcast into the blood and reach every cell, but only cells with the right receptor will actually respond. There are four properties every hormone shares. First, secretion. Hormones come from specialized glands: the pancreas, thyroid, adrenals, pituitary, and others. Second, transport. They travel dissolved in plasma or bound to carrier proteins. Third, specificity. A hormone only acts on cells that express its matching receptor, like a key in a lock. And fourth, effect. Once bound, hormones alter gene expression, enzyme activity, or membrane permeability to produce a physiological change. Chemical classes of hormones. Hormones are not one kind of molecule. They fall into three major chemical classes, and understanding those classes explains almost everything about how they behave. The first class is amino acid derivatives. These are small molecules built from a single amino acid, usually tyrosine or tryptophan. Catecholamines like epinephrine and dopamine are water-soluble and act on cell surface receptors. Thyroid hormones, also derived from tyrosine, are unusual. They're lipid-soluble and act inside the nucleus. The second class is peptides and proteins. These range from tiny 9-amino acid peptides like ADH all the way to large glycoproteins like FSH with two subunits and a molecular weight around 34 kilodaltons. Because they're chains of amino acids, they dissolve easily in blood and act on receptors at the cell surface. The third class is steroids, and all steroids share one thing: a 4-ring carbon skeleton derived from cholesterol. Cortisol, aldosterone, estradiol, testosterone, all below here. Vitamin D3 is a corticosteroid, structurally related to steroids but with one ring opened, all belong here. They're lipid-soluble, which means they can't dissolve in blood without help, so they hitch a ride on carrier proteins. And because steroids generally diffuse across membranes and act through intracellular receptors, not on its surface. Epinephrine catecholamine. Let's look at our first specific hormone, epinephrine, also called adrenaline. This is the molecule responsible for the fight or flight response. Chemically, epinephrine is compact, just 9 carbons, a catechol ring with 2 hydroxyl groups on the benzene ring, a beta hydroxyl on the side chain, and a secondary amine. Its molecular formula is C9H13NO3, and it weighs only 183 grams per mole. That simplicity is part of why it works so fast. It's synthesized in the adrenal medulla through a 4-step pathway starting from the amino acid tyrosine. Tyrosine becomes dopa, dopa becomes dopamine, dopamine becomes norepinephrine, and the enzyme PNMT adds a methyl group to produce epinephrine. The adrenal medulla is the primary source of circulating epinephrine. Because epinephrine is water-soluble, It travels freely dissolved in plasma, no carrier protein needed. The flip side is a very short half-life of 1 to 3 minutes. It's cleared rapidly by two enzymes, MAO and COMT. Epinephrine doesn't have a single target, it has many. In the heart, it increases rate and contractility. In the lungs, it opens the airways. In the liver, it triggers glycogenolysis to flood the blood with glucose. In adipose tissue, it drives lipolysis. And in blood vessels, it causes vasoconstriction in some beds and dilation in others, depending on which receptor subtype is present. That brings us to receptors. Epinephrine binds both alpha and beta adrenoceptors, all G-protein coupled receptors. Beta receptors use the GS pathway to raise CAMP and activate pKa. Beta receptors signal through GS to increase CAMP. Alpha-1 receptors use GQ to raise intracellular calcium. Alpha-2 receptors use GI to reduce CAMP. Insulin, peptide hormone. Next is insulin, arguably the most clinically important hormone in medicine, given the global burden of diabetes. Structurally, insulin is a two-chain polypeptide. The A chain has 21 amino acids, and the B chain has 30. They're held together by two interchain disulfide bonds, and there's a third disulfide bond within the A chain itself. These crosslinks give insulin its stable, compact shape. The total molecular weight is around 5,800 daltons. Insulin is made as proinsulin, a single chain, and the C-peptide connecting the two chains is cleaved out by proteases inside the beta cell's secretory granules. The zinc hexamer form you may have seen in textbooks is how insulin is stored before secretion. The stimulus for secretion is primarily rising blood glucose. Amino acids also trigger release, as do the incretin hormones GIP and GLP-1 from the gut. Sympathetic activation and epinephrine suppress insulin secretion, while incretins and elevated nutrients stimulate it. Glucagon can actually promote insulin release under some conditions. As a peptide, insulin is water-soluble and travels freely in plasma. But it has a short half-life, about 5 to 6 minutes, because the liver clears about 60% of it on the first pass, with the kidneys handling much of the rest. Insulin's receptor is a receptor tyrosine kinase. Insulin binding induces conformational changes that activate the receptor's tyrosine kinase domains. The two alpha subunits of the receptor, the beta subunits, cross-phosphorylate each other on tyrosine residues. This recruits the scaffold protein IRS1, which activates PI3 kinase, generating PIP3. PIP3 activates AKT, also called PKB, which is the central node. AKT triggers GLUT4 vesicles to fuse with the plasma membrane, dramatically increasing glucose uptake in muscle and fat. Cortisol, steroid hormone. Cortisol is the primary glucocorticoid, the steroid that governs the stress response and regulates metabolism across dozens of tissues. Let's look at the structure. Cortisol has the classic 4-ring steroid skeleton, 3 6-membered rings, and 1 5-membered ring, all derived from cholesterol. What makes cortisol a glucocorticoid specifically, and not a mineralocorticoid like aldosterone, is the combination of functional groups: a delta-4,3 ketone in ring A, an 11-beta-hydroxyl in ring C that is essential for glucocorticoid activity, a 17-alpha-hydroxyl, and a 21-carbon side chain with a ketone. remove or change the 11-beta hydroxyl, and the glucocorticoid activity collapses. The molecular formula is C2-1H3-0O5, 21 carbons, 5 oxygens, molecular weight 362 grams per mole. Cortisol is secreted by the zona fasciculata of the adrenal cortex under the control of ACTH from the pituitary. There's a marked diurnal rhythm. Levels peak around 8 in the morning and reach their nadir around midnight, driven by the HPA axis and its circadian clock. Because cortisol is lipid-soluble, it can't circulate freely. Its poor aqueous solubility would limit efficient transport and accelerate clearance. About 90% is bound to corticosteroid-binding globulin and albumin. Only the free fraction readily enters cells and exerts classic genomic effects. This binding extends the half-life to roughly 60 to 90 minutes. Once the free cortisol diffuses through the target cell membrane, it binds the glucocorticoid receptor in the cytoplasm. The receptor hormone complex then translocates into the nucleus, dimerizes, and binds to glucocorticoid response elements in DNA, directly turning genes on or off. This is why cortisol's effects unfold over hours, not seconds. The effects are broad. In the liver, cortisol drives gluconeogenesis. In muscle and bone, it promotes protein breakdown. In immune cells, it suppresses inflammation and immunity. This is the basis of synthetic glucocorticoids like prednisone as anti-inflammatory drugs. Thyroid hormones, T3 and T4. The thyroid hormones are chemically unique. They're amino acid derivatives built from tyrosine, but they behave like steroids because iodination makes them lipid-soluble. This is the exception to the simple rule that amino acid derivatives act on cell surface receptors. There are two main thyroid hormones. T4, thyroxine, has 4 iodine atoms and a molecular weight of about 777 grams per mole. T3, triiodothyronine, has 3 iodine atoms and weighs about 651 grams per mole. T4 is the form secreted in large quantities by the thyroid follicles. T3 is the biologically active form, around 3 to 4 times more potent than T4, and most of it comes from peripheral conversion of T4 by deiodinase enzymes in the liver and kidney. Synthesis occurs inside thyroid follicles. Iodide is pumped in, oxidized, and used to iodinate tyrosine residues on thyroglobulin. Coupling 2 diiodotyrosines gives T4. Coupling 1 diiodotyrosine with 1 monoiodotyrosine gives T3. The hormones are stored within the follicle until TSH stimulates their release. Transport is almost entirely protein-bound. More than 99% of thyroid hormones circulate bound to TBG, transthyretin, and albumin. This explains their very long half-lives. T4 has a half-life of roughly 7 days. Only the tiny free fraction enters cells. The regulation is a classic negative feedback loop. TRH from the hypothalamus stimulates TSH from the anterior pituitary. TSH stimulates the thyroid to produce T3 and T4, and rising T3-T4 feeds back to suppress both TRH and TSH. In target cells, T3 enters cells through specific transporters, such as MCT8, and binds the thyroid hormone receptor, a nuclear receptor that forms a heterodimer with RXR. This complex binds thyroid response elements in DNA. The effects are sweeping: increased basal metabolic rate, thermogenesis, heart rate, protein synthesis, gut motility, and, critically in early life, brain development. Iodine deficiency causing goiter is a memorable real-world hook. Without iodine, thyroid can't make T3, T4, TSH stays high, and the gland enlarges trying to compensate. Stay connected. If you enjoyed this presentation, subscribe for more anatomy and physiology lessons. Like the video. Leave your questions in the comments. Share with classmates and fellow students.